I'll do that. All right. Welcome, everyone. This is the monthly MDBA Zoom speaker series. And this evening, I'm really pleased to be able to introduce Dr. Ramesh Sagili. And I'll say a few words about Dr. Sagili and then turn it over to him for the presentation, and then we'll have time for Q&A. Dr. Sagili is a professor in the Department of Horticulture at Oregon State University and leads the OSU Honeybee Lab. He obtained his PhD in entomology from Texas A&M University, specializing in honeybee research. His primary research focus is honeybee health, nutrition, and pollination. Dr. Sagili initiated the creation of the Oregon Master Beekeepers program, chaired the Oregon Governor's Task Force on Pollinator Health, and currently serves on the National Pollinator Subcommittee. His research program addresses both basic and applied questions to improve honeybee health and nutrition, and I think that's going to be a large part of his presentation tonight. And hence, the majority of his research projects are collaborative efforts involving stakeholders, that is, both beekeepers and growers. He has authored several important research and extension publications. OSU Honeybee Lab is at the forefront in critical honeybee nutrition research, and the Oregon Master Beekeepers Program is one of the among one of the top bee extension programs in the nation. Dr. Sagili has received several awards, including the Entomological Society of America's Pacific Branch Research Award, Eastern Apiculture Society's Outstanding Research Award, OSU's Excellence in Postdoctoral Mentoring Award, and the OSU Outreach and Engagement Award. It's my pleasure to welcome Dr. Sagili. Thank you very much for joining us.
Thank you, Ian. Can you all hear me well? How about my nice English accent? That's good too. Okay. Okay. So no, it's a pleasure to be here. I know. I don't think I have I spoken to your group before, Ian, or is it first time? Yeah, you said some of your folks they heard me at the American Beekeeping Conference, I guess. Yeah. Yeah. So good to be here back. Yeah. So what I'll do is I know Ian asked me to focus more on honeybee nutrition, but I decided to talk a little bit about Varroa as well. I know I can't really do a talk without talking about Varroa mites. So I have snuck in some of the slides there as well, but I'll try to spend more time on nutrition, but or maybe half and half. But if you guys are bored with the Varroa, you know everything, then probably we can just talk about nutrition. But Varroa, as you know, those two things are really important. Both Varroa and nutrition are really critical. And it looks like you all are very experienced as well. Just to know, there are not too many people in the audience. I can see all of you. Just with the raise of hand, how many of you are very new, like just started this year or last year? So looks okay, just one of you. Okay, perfect. Yeah. And others, I'm assuming you have at least 3, 5 years experience minimum. Okay. And how did the bees do this year? Most of you, they survived the winter. Okay. Yeah, I know you probably heard, right? Commercial beekeepers, they had a big problem. So at least there is the largest beekeeper in the nation. His name is Brett Ady. Ady Farms. I think they move bees between North Dakota for your almonds in California as well. And so they lost probably 70% of their bees. So that made a big national news, right? So you saw this Project APSM, which is a nonprofit. They did a survey and they are estimating that probably if you put all these big commercial beekeepers in the nation together, they lost about 60% of their colonies. But that's not with the case with hobby beekeepers. Even in Oregon, I do surveys for hobby beekeepers. They have lost probably around 30% max, I would say, some of them, but no one has lost 60%. That's a big number. But commercials, yeah, some of them have lost at least 50% in Oregon as well. So last year was pretty bad for some of the commercial beekeepers. Again, several reasons. But I'm glad the backyard beekeepers didn't go through that pain this year. So yeah, it's always a challenge, right? You see, it just keeps ups and downs of these colony losses. It's not very consistent. Okay, you want me to share my slides, Jan? Okay. And I'm notorious. I can keep you here until midnight, but I'm not gonna do that. What's should I stick to 45 minutes or and then questions or an hour and then questions or
45 minutes and then questions or an hour. I mean, it's fine. We usually have
okay.
questions. We usually have questions until about, 8:15, 8:30. So that's fine. I'm a teacher as well, and I usually say to my classes at the end of an hour and a half, you guys are okay for another 4 hours, right? We can just keep
Right? Yeah.
going. So
Yeah,
yeah. Yeah.
yeah. I love talking to you guys. So I can, yeah, I put a lot of slides you'll see, but stop me if you think you guys are tired and you don't want to go any further, I can stop anywhere. But please feel free to ask questions as well. If you think there is anything pressing, yeah, just unmute yourself and ask. But yeah, I can spend some time at the end as well to answer some of your questions. Yeah. Okay, let me share my screen. Let me go back again. Yeah, I have some audio as well. So let me make sure that I have clicked audio as well. Yeah, let's see if we can't hear the audio on some of the videos, then let me know. I can go back and change. Okay, so my lab actually, I have been at Oregon State University for about 16 years, and we do focus a lot on honeybee health. So when I say health is a very big term, right? So we look for anything from Varroa, Nosema. We've done a lot of work on Nosema, a little bit on viruses, more on bacterial disease. We have a huge project going on right now for European fall brood. I know it's not a big problem for backyard beekeepers, but some beekeepers that do blueberry pollination, especially. I know California doesn't have huge blueberry acreage. I know there is some, but not huge. But Michigan, Washington, Oregon, these are big states that grow blueberries. And when beekeepers go to blueberry pollination, the commercial guys, they always report a high incidence of European foul brood disease, which is a bacterial disease, as you know, and it doesn't go away. Usually it's a spring disease. We used to think, it goes away by June or July when the nectar flow starts. But these days we see it persists until August, September as well. And so we are looking very deeply into that. So actually I come to California when almond pollination is there. We do a longitudinal study where we are tracking about 1,500 hives from Washington, Oregon, Mississippi, and California as well. So that's a place where we start in almonds. That's first cycle for pollination for most of the beekeepers in the country. And then those tagged hives, they move to back to their own states and then they do blueberry pollination. So we take these longitudinal samples. And so we're trying to see if there is a new variant. Like we are tired of all these variants with viruses, right? With especially COVID. We heard from Delta to Omicron and so many. So bacteria also mutate, right? So then we probably are dealing with a new variant. We don't know that yet. So we're trying to take samples and do some genetic analysis as well to see if, because probably last 15, 20 years, no one has really looked at any changes in European foul brood bacteria. So we're looking into some of those and other factors that might be responsible for the spike in European foul brood disease. So we do a lot of other things related to health, but nutrition is very near and dear to my heart is because that's an area people think, we understand bee nutrition, but you'll be surprised when we say we know more about grasshopper nutrition when compared to honeybees, because those are easy to study. Whereas honeybee being a very complex social organism, there are different levels, right? We are talking about a colony level nutrition, then you see your larvae is being fed a different diet, right? That is produced by those hypopharyngeal glands and mandibular glands. Whereas if you are a grasshopper, you lay eggs and the nymphs hatch, right? And they start eating the same thing the mother was eating, the same plant. As in your bees, think about the complexity of the diet, right? This is a totally different diet that is being fed to the larva. So again, just to appreciate the complexity of nutrition, it's not that simple to study nutrition in bees. So people have, that's one more reason why honeybee we do understand. I'm not saying we don't. Honeybee has been the one of the organisms that had been studied for more than 200 years in a proper setup, right? So, but still, there is a huge gap in knowledge, especially micronutrients. And I'll talk about that in the second half. So I'll start with Varroa, then we'll move to nutrition. Okay, so before I start, I want to always I put this slide in the front. I lead the honeybee lab here, but without all these wonderful people that you're going to see in the next few slides, it's impossible to do what we do here. We have like probably 10 projects in a year, and with all this, so like this slide is these are all existing ones, but then I call some drifted bees that have gone and taken different positions, like Cameroon Jack. You see the fourth one on the top. He's a assistant professor in University of Florida. He was one of my students here, like probably 10 years ago. So people have moved on and are in the bee industry as well. And these are some other undergrads. I don't keep up with all of them, so probably they are doing something different than honeybees at this time. But again, all labs, it's not just me, right? All labs, we really appreciate the services that undergrads and graduate students and postdocs provide. And without them, any lab will not be successful. Uh, just again, a raise of hand. How many of you use oxalic acid for Varroa treatment? Based on that, I will see if I can. Okay, I can't see everyone. I see one hand there. Let me scroll down. Okay. So, yeah, oxalic, we have done few studies in the last few years, so I'll be quick on those. But if you have more questions at the end, I can answer. And oxalic is becoming more popular, right? I know this product has been used even in the US for several years now, and Europe, they have been using it for 30, 40 years probably now. So it's an interesting product, and I know again there are new products coming on. New means in the sense of formulations, right? So I can talk a little bit about those as well. And then I'll go into honeybee nutrition. Okay, this is a slide I always put. Colony survival is dependent on two important things. I'm not saying these are the only two. There are several other factors, but these are the prime factors. If you can somehow control Varroa in time, and then the nutrition, I know, which is not so much in your control sometimes because it's all climate dependent. With at least Varroa, you can do samples and you can figure out what your thresholds are and you can do something about it. So these are the two important factors, especially I think one of or two of you are new beekeepers in the audience. So always think about these two. These are really critical factors. These are two dependent. If you have taken care of varroa and nutrition to some degree, probably there is a 90% chance that your colony will survive the winter. But still, there may be 10% which is beyond your control, I guess, all the time. So here, let me move the top. I think you can't see that one. Is the top bar coming in your way, right? You can't see the top part where I say Varroa mite life cycle. Are you able to see that or?
We're able to see that.
Okay, perfect. Okay. So it's maybe just on my side here. I just have to say, okay, okay. So there is two phases, right? Again, you all are experts and you know the Varroa life cycle very well, but I'm going to ask you some questions probably to just test your knowledge on Varroa. So we talk about phoretic phase. I know some people don't like to use phoretic phase. There are some definition issues, but we'll forget about that for now because the literature still, most of the literature is still mentioning them as phoretic phase. So when I say phoretic means what you see on the left, right? The bee has all those mites. So it's basically, I try to be more specific here. Maybe I'll say a non-reproductive phase. That's a phase that is feeding on the bees, right? And then on the right bottom, you see those are that's a capped frame. You'll see if I ask you how many mites are there inside those cells, probably in your hive today, probably you can't tell me, right? Because it's hard to guess what's there inside. So we'll talk about that in a second. So let's finish the life cycle. And just for those, bear with me, those who are veteran beekeepers here and know much about Varroa. Uh, so the life cycle is really critical. I know we think always, oh, we know everything about Varroa life cycle, and but it's really important to really refresh your memory always about Varroa mites. So there is a reproductive phase, there is a phoretic phase. Let's talk about the reproductive phase. So the female mite enters a cell when the larva is about 12 hours to be capped, right? The capping happens, and then you will see that they start coming out of those brood food area, and then they start feeding on the larva, and then they start laying eggs, right? So that's what I say here. Once the larval food is consumed by the larva, the mite gets out, and then you see the feeding happens, and then about 2.5 to 3 days after cell capping, the first egg is laid, which is a male, and then subsequently she lays about 30 hours, every 30 hours, another female egg. So she may lay about 5 to 6 female eggs in her cycle in that cell. So the male and the first female are ready about a week after they are out from hatching. So watch this animation if you didn't understand anything that I said. So here is a hive that you see a bee with a mite, a female mite that is mated, right? So she enters the cell. So they are using this pheromone cues. They know the larval stages. They can easily detect which larva is 1 day or 2 day or 3 day or 4 days. So they are entering a larva which is about 12 hours behind in capping. And then she starts laying eggs once it's capped. And then you see those females coming out, hatching, and then there's this inbreeding, right? Probably you all know that the male mates with the females that are produced, which are his sisters. And that's another question, right? Inbreeding we all think is bad, but then isn't it very amazing that the mites have been very successful even with their inbreeding process? Again, that's what I said. I can keep you until midnight if you want to go into those things, but don't ask me about those today. Uh, but yeah, it's very fascinating biology of the mite. So that's how you see a female parent mite that went in, came out with one offspring. That's usually happens if it's a worker cell. But if it's a drone cell, I know sometimes we may see two offsprings come because the drones take 24 days, right? That's a life cycle. Worker is 21 days from egg to a worker to come out of her cell, whereas drones take 24 days. So even 3 days window makes a big difference for their maturation and mating. So that's why you get two offspring. And have you ever thought about what happens to the other females that I talked about, right? She laid about five or six. So they all die because some of them are not sclerotized. You see, when I show you this, let me go back again. You see these mites, they're pretty brownish or reddish brown, right? That you see. So that's called a sclerotization process. So the chitin has to be fully formed. And whereas sometimes you might have seen, if some of you use sticky boards, you see some mites that are pretty pale color, right? You see on the bottom boards because they're not fully mature enough. They died inside those cells and the bees removed them and they are falling on your sticky board. That's why you see some whitish or little more pale colored mites that you see. So here is a study, probably you all are aware of this, right? Samuel Ramsey, he did some work on figuring out. I mean, this was known that bees probably are not, the mites are not just feeding on the hemolymph, but their diet probably is different because of their mouth parts. I think the theory was there for, the hypothesis at least was there for a while, but no one had really tested it in a more controlled way. And so these guys did a great job of publishing this work where they are showing that most of the times those mites are hiding in the ventral side, right, on the abdomen. And most of these, you see that red part that you can see on the left, if you see my cursor. So they found about 60% of mites in that area. Most of those mites were there. So they're feeding on those abdominal fat bodies and fat bodies, those of you who are new to mites, they are like your liver, right? They serve that type of function for immunity, their metabolism, so many functions. And if you're compromising their abdominal fat bodies, then those bees are no good. So that's why when you can't control varroa mites, especially when they are making those winter bees or diutinous bees, I'll talk about that in the probably in the nutrition section. So those really are important. I know California, you guys don't get a real winter depending on where you are. So your brood is already there in December, January, a little bit as well. I guess there is no really broodless period probably. So that's what happens. These mites are all waiting for the brood, right? So and they are on these adult bees feeding on the abdominal fat bodies to survive the winter. If you live in Minnesota, probably those mites are living almost 5 months on the bees. So let me ask you a question. What's the life cycle of, I mean, not like, I showed you the life cycle. What's the lifespan of a mite? Have you ever thought of how long the mites can survive? You can unmute and answer if you're curious. What's the lifespan right now? If you have mites in your hives, how long do they survive? If you paint a mite today, you go and catch a mite somehow and put a green dot or a blue dot on that. It's a painful exercise. I'll show you some of the work that we do with those. But how long do you think the mites that are right now in your hives would survive? Okay, maybe Ian doesn't have any door prizes, so probably no one is answering. That's okay. So
Maybe a few months. Up to a
Uh, yeah, yeah, maybe not few, but at least people have shown these are very, very painful. I know the answer I'm gonna give you is not a very robust one either, but I have seen couple of studies, very painful, painstakingly they have done because you have to. How do you know the life of a mite, right? You have to somehow put a marker on the mite and then in that huge 40,000 bee colony, you have to find that mite back again and see how long it survived in the hive, right? So these are really impossible sometimes to do, but some people have done those type of work. And so usually we think the mites survive about 40, 45 days in spring and summer when they're really productive. going back and forth reproducing, because it takes a lot of toll on them, right? If they are continuously reproducing, their physiology is very stressful at that time. So that's why they survive only about 45 days, I would say. But think about the mites that are on your bees from November on. If you live in Minnesota or some more temperate climate, they are on those bees for almost 5 months. So it depends on the time of the year, right? So if they are in winter time, they can survive 4 or 5 months. If they are in summer, they are continuously actively reproducing. So there is a lot of wear and tear on them. So that's why they survive only about 45 days. So that's basically what we think the mite lifespan is. And then have you ever thought how long a mite will wait before she gets into the cell? So I showed you in that animation, right? You saw there was a parent mite that went in and then there was an offspring. When do you think these guys will get back into another cell to breed? Any answers?
Just before the cell is capped, there's a pheromone that the larva sends out.
That part, yeah, that's a great answer. But I'm asking how long they will wait. So if a mite, so those mites come out of the cell, right? The parent that went in, she just came out of that cell after the bee came out. Will she go immediately into another cell to breed, or will she wait for some time?
Immediately.
Immediately, okay. Any other answers? Yeah, people have shown some mites in the lab, they have the ability, like Ellen said, yeah, immediately. Some people have done experiments in the lab where they have put the mite as soon as it comes out of the cell with the bee, they have put in another cell and they were able to reproduce. But in nature, generally what happens is after the reproduction, they have to rejuvenate back, right? They have to gain their physiology back. So usually they take about 5 to 7 days, they feed on the adult bee before they get back into those cells again to breed. So that's basically their physiology. But yeah, as Ellen said, yeah, you can they have the ability. If they want to go, they can go next day or same time. As soon as they come out, they can get immediately as well. But most of the time, what the people have documented is they take about 5 to 7 days to get back their physiology, or because it's a stressful thing, right? They're going inside and laying eggs, and so there is a lot of physiological stress on them, the reproductive physiology. So they have to get back to shape and again start laying eggs. So they may wait. That's the reason when someone tells you that you have to use oxalic acid 5 days apart or 7 days apart, some of that has to be related to the mite life cycle as well. That's what I'm giving you all these things is you can think about it. Why am I using oxalic every 5 days if I'm doing a vaporization method? But if you're doing some oxalic glycerin type or something like Varroa sand that is being sold now, then that's a continuous release. That's a different story. But I know there are a lot of beekeepers that they do vaporize, right? You burn that 2 or 3 grams at the wand. In that case, you need to understand the biology of the mite as well. These are symptoms I don't have to go through. You see deformed wing virus B here, right, on the bottom, and then there'll be a lot of other problems, not just the physiological stress that the mites create, but the major damage they do. And sometimes as beekeepers, we forget about viruses. The viruses are the ones that do the final damage, right? So yeah, I'll talk about viruses in a second too. But here I have another question for you to think about. So here I'm asking you, there are mites inside those capped cells, right? So if I ask you go into your hive today, and then you may have several frames of capped brood probably. So what do you think? How many mites are inside the cells, and then how many are there on the bees? So if you have 40,000 bees, I'm just making up some numbers. If you have 40,000 bees in your hive today, I'm asking you how many mites would be on the bees, and then how many are inside those capped brood frames if you have 5 frames brood or 7 frames brood. So think about it. So is it 50-50, 80-20? Just think about an answer and I'll show you what we found in a painstaking study we did here in Corvallis, Oregon. Uh, uh, Probably 7 years, 6 years ago. Yeah, probably. I'll show you the data.
20,
So
2080.
20-80. So you're saying 20 in the cells and 80 on the base?
No, the other way.
Other way. So that's then you're talking about B. Okay. So you're saying 80 in the cells and 20 on the base. Yeah. That's a good guess. But yeah, there are probably 3 or 4 correct answers as well here. It's not just one. And it depends on the time of the year. So that's another trick as well, right? I didn't tell you which time, right? So if you have very less brood in your colony, then it's a different answer, right? So I'll show you a study we did in September. And again, I'm not looking for one solid answer here. I just want the new beekeepers in the audience. I know the ones that we experienced here, probably you have to deal with the mites inside the cells at some point as well. But sometimes I get these questions from new beekeepers that started beekeeping maybe a year or two, and they would say, oh, I was asked to use oxalic acid vapor. I hit them with oxalic vapor in June or whatever, July, and then they think all the mites are gone because they don't think about the mites that are in the reproductive phase, right? Because every day, if your queen was laying 2,000 eggs in a day, and for example, if they're 50% infestation, so there might be 500, sorry, 2,000 bees coming out means 1,000 mites coming out as well. So who will deal with those in the next few days, right? So that's what I'll come back to that in a second. So this was just a question to just show you that importance of thinking about mites inside the reproductive cells as well. So this was a study with the population dynamics. We did, I think 2018, I believe we did this. So you see on the far, so this is all forested area in Oregon. And you see there's a lab truck and there are students you can't see very well. So we chose a very isolated area because here we wanted these 6 colonies that we were using, and we were not treating them for a while. So we didn't want to spread mites to other beekeepers around us. And so yeah, instead of breeding for bees, we were technically breeding for mites in this case, right? Because we wanted to see the mite population grow with the bees, right? Without having any miticide inside. That's how you have to figure out the mite dynamics. If I'm using a miticide, then I'm messing up with their mite life cycle, right? So that's not the way you do it. So here is the study. Technically, you may think, 6 hives look small, but you'll see even 6 hives is a big pain because the way the study is done, you will see how much work it is to do. So we didn't treat the hives from March until September. Those are the dates I'm showing you there. And then we went in September, on September 14th, and we killed all the colonies, right? How do you kill a colony? Have you ever thought of it? I know we can easily kill our colonies by not attending them and not doing anything for miticides or anything for Varroa, and then also for nutrition. But if you want to experimentally kill colonies, so this is how we do. So we shake all these bees with this funnel into a tub that you see on the bottom that has alcohol in there, isopropyl alcohol, ethanol, whatever you want to call. And so we put all those bees in there. I know you can't get 100% of the bees, but so we chose a day in September here in Corvallis as well. You can't find a day that is cooler. So we went around 6 o'clock or 6:30 around and tried to put as many bees we can. So you see some bees are still basking on the students here on the back. And so if you want to still see the video, let me see video. Are you able to hear the background noise on the video or no? No, okay, then I think I have to see. Yeah, watch this video. I don't think there is much here, but then a couple of videos have some good background music. So I'll play that when it comes. I think I have to choose an option there. So one of the students is playing with the brush there. Huh? So we didn't pay her for that day. So what we are doing here is we're shaking all the adult bees into those buckets of alcohol, and then all the brood frames, they go back to the lab into a freezer. And then the students will painstakingly sit. So these are the buckets full of bees that back to the lab. And then there are thousands of bees, right? So we have to have a method where we do a subsample of those bees and then count mites on there. So we have a mite shaker. And so yeah, we do a lot of different things in the lab, which you don't need to know. So this is a painful process. You see the students looking into each of those brood cells for mites. That takes a long time. And as I said before, we probably do 8 to 10 projects in a year. And this was just one. So we took almost 2 years for them because they were taking sometimes whenever they had time in between the other experiments. So finally, yeah, this is what you see. They are pulling all those pupal stages or larval stages out, and you see the mites on them. So that's how we count for those mites and the mite families in there. And so, yeah, it's a little hard to find. So these are all young undergrads, right? So their vision is pretty good, but still we gave them those magnifying glasses with the visors on. But it still took a long time. And none of the students have sued me now for loss of vision. So I think we did okay with that project. So you see here, these are not different mites from different colonies or different. So this is all one family. I don't know if you ever looked into a mite family. If you have a good drone brood, sometimes you can pull one if you have enough mites in your hives, and you will see if they are in this stage, you may find all those. So on the right, probably this is the male, and then these are all the females that are not fully mature. So probably this one, if you're able to see my cursor, that's the female that entered the cell, the parent mite, and this mite on the far left top probably is the one that will come out with the mother because she's ready. But all these white ones I talked about before, these will die inside those cells. And that then the bees will remove them. And here you see another bee, like a almost fully formed bee that we pulled out, and you see that mites here. This is all one family. So there is a mother and then there are offsprings here. So here is the answer for you that you are waiting for. So we found about 56% inside the cells and about 44% on the bees. And that's very specific to here in the valley, where the Willamette Valley where we are. Probably beekeepers can think of this. But again, it depends on where you live. But again, the take home message, what I have for you, especially the new beekeepers in the audience, is you have to always think about mites. At least there will be 50% for sure, and even more, depending on the time of the year. If you have more brood, yeah, expect more of those bees, and then more of the mites getting in. And sometimes if there is less brood, then you'll see multiple mites getting into the same cell. That's a bigger disaster as well. Then those pupal stages or larval stages, they may not even survive sometimes. They may be very bad. So the bee may even not come out if there is multiple mites, parasite is in just one larva. So it depends again. That's what I say here. The take home message is more than 50% mites are in the capped cells during brood rearing season. And when you talk about November, December, probably 90% or 100% are on the bees. So keep this in mind, especially the new beekeepers when you do treatments. So you have to think about the mites, not just one treatment, because if you don't think about the biology, you may think the mites that I kill on the bees is enough. No, it's not. You are just killing the bees for that day that was on the bees if you are using a vaporizer. But what happens to the bees, the mites that come next day, like another 1,000, another 1,000? I'm just making some numbers. So you have to think about that. That's why when we use oxalic vapor, we ask you to choose a 5-day interval or a 7-day interval so that you can get rid of most of the mites that are in the colony, because there will be mites. So if they're waiting for 5 days, right? Suppose today you went and did a vaporization. So you kill the mites that are on the bees, probably 90% of them, or even sometimes oxalic is very effective. You can kill even 95% as well. And then after 5 days you go, hopefully the bees that would hatched in that last 5 days, they're still not entered the cell. So you can hit one more time, right? And then you can do, so you can do maybe 3 times, 5 days apart, and probably you will get a good control with the vapor. So I briefly mentioned about viruses. Usually as beekeepers, we don't tend to think about it. And I know you can't do much with viruses, but at least it's a good thing to understand. The mites do a physiological damage by feeding on those abdominal fat bodies and a little bit of hemolymph. But then eventually these lethal viruses are the ones that are responsible for the decline of hives. You probably heard of deformed wing virus, right? And I don't know if you have heard of, there is a deformed wing virus A, there is a B. They're talking about C and D as well. And I don't know if you ever heard about recombinant viruses that I have listed here on the bottom. So recombinant, for those who have never heard of this term, is so when there is a deforming A and B, there might be genetic shade between those two, and there is a third one which may have half the genetics of A and B. So again, I'm giving a very layman terminology here. So those are recombinant viruses. Sometimes recombinant viruses become more lethal than the original virus. So those are the things we still don't understand. And there are only few labs in the country that do viruses. We do, but again, I'm not very well equipped. I'm not a virologist. I'm a honeybee biologist. So our knowledge is still limited. So I wish there are more people with virology background that study bees, then probably we'll have a better handle on viruses. Again, We can't do much, but at least we need to understand the phenology of these viruses. And most of these viruses are connected to Varroa. So Varroa is the carrier. There are a couple of viruses where you don't need even a mite. The bees can transfer among themselves with trophallaxis, which is exchange of food, or they can defecate, and then if the other bee accidentally touches the feces and then ingests or something happens, they can get the. So, but again, most of the time it's the Varroa control. So that's what we tell people, that we can't do much with viruses, but if you can control the Varroa, then probably the virus titers will be low in your colonies. Any questions here? I know probably some of you are waiting for answering, but yeah, if you don't, then I can keep going. So this is nothing rocket science, you all know, so I'll not spend much time on this. I just put something for new beekeepers. So there are different ways to sample. Sampling is really important. I hate giving numbers. I know if you read books, people will say, oh, or you read that Honeybee Health Coalition guide and all. They're all nice. You need to have some number in mind, but again, don't rely too much on the thresholds. Like sometimes we say, oh, 2% is a threshold, 3%. Those are not magic numbers. Like I talked to some beekeepers here in Oregon. You will laugh if I say that some of them will say there is no threshold for us. 0 is our threshold. We treat them meticulously every other month or whatever their schedule is, because mites are always there, right? It's just that you don't want them to get to a level, but that doesn't mean that you have to randomly use miticides all the time, because most of the miticides are toxic to your bees to some degree as well. Nothing is safe. Think about miticides as they're relatively safe. I have not seen one miticide that is totally safe for bees, right? So we always talk about relative toxicity, and that's what we should do as well. It's not so like any synthetic miticide like amitraz. Some of you probably use that APR. So those are synthetic miticides. So if they are probably 100 times toxic to mite, they probably still are 5 times toxic to your bees as well. So nothing is really safe in terms of that's a challenge with my bees. People think, oh, why are we not having too many miticides that help beekeepers? Because designing a compound that will be safe. So I know these mites are not insects, right? But your honeybee is an insect. So, but still people use this very layman terminology, bug on a bug. So how do you control a bug on a bug, right? If something you're trying to kill, that will also impact most of the systems are similar. So you're spraying or using a chemical to kill mites, then it will impact your bee as well, even though they have tried to make it very specific for mites as well. So these are some methods. I'll not spend too much time, but if you're interested, I can send you some links to watch. We have several of those methods on our website as well, if you're interested to go and look for. So here is a quick strategy. I'm saying frequent monitoring of mite levels from spring to late fall is important. So calendar-based treatments are no good. I know if you talk to beekeepers that have kept bees for 30 plus years, they'll say at some point they used to use calendar. Okay, Labor Day you do this, on Memorial Day you do. I don't think your mites and bees are following any of those calendar days. So you have to look for the brood rearing. If the brood rearing starts early, then your mite treatments have to come earlier. And that's what I say here. Early brood rearing means early mite control strategies. So here are some methods that I mentioned before. I'll skip that one. And always try to use combination of miticides. I know some of you probably dislike miticides. You don't want to. I don't blame you for that. But unfortunately, we live in a world right now where without treatments, I don't think you'll be a successful beekeeper, because Varroa is a big menace. So you have to monitor them and at least do something. If you don't like synthetic chemicals, but there are some a little more softer chemicals that we call organic acids, like your formic or oxalic or APY, sorry, the other one, AP guard, which is thymol. There are other options as well. I'll talk about them in a minute. So here is something that we have used, like amitraz during spring, then formic acid during summer and fall, depending on the temperatures, right? These are all temperature sensitive. Amitraz you can use probably even during high temperatures, but not during nectar flow. That's a synthetic chemical. Then you have formic. Some people use only one pad and then replace it after 10 days. That works as well. Or you can use 2 pads for 14 days. So always read the label and use those according to that. And we still use oxalic dribble. I don't know if you guys do this, but oxalic dribble in winter. So it's a dribbling method. You make a 3.5% oxalic solution in sugar, and then you dribble on the seams like 5 milliliters, so that it falls on the bees, and it's attractive because there is sugar. They'll groom themselves, and then it gets to the mites on their bodies. So that's one way, and it's pretty effective actually. There are several beekeepers in Oregon and other locations, they do oxalic dribble. So that's probably once you have done all the control in fall and summer, and then if there are still some mites remaining, then you get rid of them, and because you will have less mites to start the next year, right? That's really helpful for your bees. Always monitor mites after treatment, because sometimes the treatment may not work. And then if you think my treatment worked and after 2 months you're going back, the damage is done. So always monitor mites. If you do an alcohol wash or whatever your method is, then go back after 3 to 4 weeks and do one more alcohol wash to see if the mite numbers have changed. So here is another example. Again, don't take this very seriously and start using in your area depending. I'm just saying what works for us here in Corvallis area. So we use APR, Amitraz in spring because the mite levels are low at that time. So Amitraz is a good chemical. It's a slow release 3.3% Amitraz. But if you use APR when your mite levels are exponentially increasing in late fall, maybe that probably will not get the mite levels back down in time. So the brood damage might be more, and your bees that are produced at around that time as winter bees, they might be impacted, and their lifespan is really important, right? The winter bees that are produced in late fall, those are the ones that survive the winter. So you don't want those bees to have in compromised health with mites or with nutrition. Any questions? So here is a list that EPA has, and this is unfortunate. See how many are listed, and people may think beekeepers have a lot of options for Varroa, but unfortunately all the yellow ones here, they have gotten resistance, so no one uses them anymore. And the green ones probably are the ones that you guys have options right now. The AP-Bioxol is nothing, it's an uroxalic acid. There are Mitaway quick strips, these are two formic acid products. This is a thymol one. I don't know if you ever used HopGuard. It's a good product as well, but again, you have to use multiple times. And the company hasn't done a great job of giving good information on it. So some beekeepers don't use this one, but that's a good product as well. And this is your Varroxan. Has anyone used this one? It's an oxalic strips that have come in the last year or so. So it's a slow release one, not oxalic glycerin. It's a fiber stuff, which I'll show you in a second here. This one. So they are like this fibers. I think they impregnate with oxalic acid. So they call them slow release strips. So I have heard some good things about these. So if you have not used them before, I know they're a little expensive, but if you think that's a good option, so don't expect that they will just get rid of all mites immediately and you'll be all clean. I think it's a slow release product as well. So you will see the mite levels will be consistently low, but they will not go down like crazy down. So you do still have to monitor and take maybe some other steps as well, maybe use formic after this and not just rely on just one product. And there is one more product that EPA is probably going to register this in the next year or so. They say it is promising, but we have to see. This is called a double stranded RNA. I don't know if you have heard of this. So it's basically a double stranded RNA that is fed to the bees, and the bees will get into their system, and then, or they feed the larvae, sorry. And then the mites that are, so it impacts the reproduction of mites. It's not technically killing the mites on the bees, but it's killing the mites that are reproducing inside the cells, because the larva will feed on this double stranded RNA, and then that product will get to the mites, and that it impacts one of the genes responsible or the proteins responsible for reproduction. So that's how it works. It looks promising the way it is described, but again, delivery is a challenge. So we'll see once it gets to the market, and then we will also do some studies here at OSU and look at the efficacy of the product. But yeah, if that works, I think that's a very good product. If it will not have much impact on the bees, it's relatively safe, not relatively, it's probably more safer than any other products that you have used so far. Okay, it looks like I'm going too much on these things. So what I'll do, and maybe I'll skip the oxalic studies and come back, or if you think, if you guys really want me to go into oxalic, then I'll go, or else I'll switch to nutrition. What's the majority opinion? Are there anyone that is obsessed about learning about oxalic?
If you
Oh,
could just
okay.
give a quick summary, maybe.
Yeah, okay, then I'll do this in 2 minutes then, okay. So we did several studies in 2019, 21, 22. I don't know how much dose you use, but the label says 1 gram, right? 1 gram per brood chamber. If you have 2 boxes with brood, then you are burning 2 grams. But that dose is not adequate. That's what most of the studies have shown, including ours. So we submitted a manuscript like a few months ago. It's almost ready for publication. So I'll show you some of the results from that study as well. So 1 gram doesn't work. So we did several studies to prove that 1 gram is not the right dose. And so we tried 3 and 4 grams. To give you a very quick recap. So why we use 3 gram is 2 gram also is not very different from 1 gram. So again, remember 3 grams per brood chamber. So if you have 2 brood boxes with brood, then you're burning 6 grams. So we tested 6 grams, 2 boxes, 8 grams, 2 boxes, right? And these were 1 week apart. And then we look at all these different parameters that usually we measure. So this is how we do, like you have to use 2 wands at a time because each one only burns 2 grams. So you have to put 4 grams at the same time so that you're not confounding the study. So I'll not play this video. I'm just going to show you the results. So we put sticky boards to see how many mites fall because beekeepers are curious. How long does this vapor work? Does it work for 3 days, 4 days? That means, does it keep dropping mites that far? So what we have done is here is after the first application, we are looking here, right? After immediately after the application, within 24 hours, you see the maximum drop happens. About 4 gram one here, you are dropping about 1200 mites here. 3 gram, and then this is your control. So after 2 days, that means 48-hour data, you see still some drop. It's not like the first day, but you still get some drop here between day 1 and day 2. There are still mites dropping, but after that, we don't see much going on. So this is your second one here. You see, this is what I was saying. For new beekeepers that are confused, I used oxalic once, and why am I seeing still mites? You see, those are the mites that are coming from the brood, right? Next day. So if all my mites were gone, then why am I getting again more now? I'm getting about more than 1400 mites falling in day 1 here. And then that it goes down, and then you see by day 2, there is not much happening after that. After the third treatment, you still see even more mites dropping. Here you're doing 2000 plus for the 4 gram. The green is your 3 gram. That one, right? So we also looked at the toxicity to larva because that's more important, right? If you keep increasing randomly your dose, what happens to your brood, right? You have to be cognizant of that problem as well. So you don't want to impact your brood. So it looks like, again, I'll be very quick here. So you see that is a 4 gram. So I'm talking about mean number of healthy larvae, right? So healthy larvae are lower numbers here, but then there is no significant difference between your control and 3 gram. So it looks like 3 gram is doing a good job compared to in terms of larval mortality, there is less, whereas it is on par with your 4 grams as well. Even the 4 gram probably still giving you a better control. So here is a take-home message from that study. So 1 gram doesn't work. So you have to use 3 grams. That appears to be more effective and relatively safe to larvae and your bees. So that's again based on our study, but I'm not EPA. The EPA label still says 1 grams. I'm not suggesting you to violate the label, but probably off the record, you can still, as beekeepers, you probably can try. There are beekeepers using it, but hopefully the studies that we are publishing and some from University of Florida, then EPA may change the label. I don't know why they chose 1 gram. It still baffles me. 1 gram is a very small dose for 1 brood chamber. It's not effective for controlling mites. So we have done some work with amitraz resistance. Maybe I'll skip that for now. You can just see pictures. So amitraz is a product that is widely used by commercial beekeepers. Off-label, you probably know. And then the registered product is the one we are testing here to see if it is creating any resistance problems. We are seeing some. It's not like total disaster, but we are seeing pockets of resistance with amitraz. So we're doing a vial bioassay. You see those mites in a vial that is coated in the bottom here with a pupa in there. And so we put 10 mites in different concentrations, and then we watch them for 24 hours in an incubator. to see how those concentrations are effective on control of mites. Okay, just for fun, I thought after dinner you probably are sleepy, so I put some fun stuff here. What is this behavior? Have you seen this before in your hives? What do you call this behavior? Can you say that again? Washboarding behavior, right? Have you heard of that term before? Yeah, people say washboarding, so just like cleaning. I don't know why they coined the term washboarding, but sometimes it's not it doesn't happen. I don't think probably some of you have seen this before. So in the afternoon, sometimes you'll see these bees, they're all facing down on that, right? And they do this back and forth motion, rocking motion. And so some people think they're cleaning the hive outside. What is there to clean outside? Some people think they are cleaning their mandibles. And so there are a lot of hypotheses, but still as researchers, we have zero understanding why bees do this behavior. So you still have a chance of winning a Nobel Prize if you are trying to figure as a beekeeper what this behavior is. I have a video. Let me see if I can play them. Are you guys still okay? I know I'm running out of time here, but I'll go quickly on the nutrition in a second. Let me stop sharing, and there is some music behind this one. Was the sound playing, the background music? Okay.
Yes, it was.
Okay, perfect. Yeah, so that's what we think. It's no one really knows. So there are a couple of hypotheses, as I mentioned. So you see, they don't even touch their forelegs on the thing. It's just like back and forth. They're not touching their antenna much, and so it's just really baffling why they do it. Some people think they are just cooling off outside. Why would they cool off outside, right? It's just doesn't make sense. So again, very interesting behavior. I'll show you one more video, and then I'll switch to nutrition and finish in 10 minutes probably. Have you seen queens fighting before? And this is a behavior that is pretty natural, right? In a hive, when swarming happens, what happens? They make about a dozen queen cells, right? And then probably sometimes two queens may come out of their cells at the same time. They find each other, they fight, and then one survives, the other gets a lot of stings and dies. So that's basically your biology. So here again, just for my class, I teach a honeybee biology class. I do a lot of research, but I have a teaching appointment as well. So for classes, we sometimes make videos. So these were two queens that we bought from one of your California producers. And then we put them together on a bench, lab bench. That's what you're seeing. They're on a lab bench and then they fight. So you probably have heard that the virgin queens always fight, right? But in this case, these are two mated queens. So that's not virgin queens. Queens can fight anytime you put two different queens in the same place. So I want you to watch one very peculiar behavior, which you probably have never seen in any other videos, which happens in this one. So I'll play this very quickly. Watch the darker queen. Keep watching the darker queen. Something will happen in like few seconds. The one is a Carniolan, the lighter one is the Italian queen. You probably figured it out. Watch the darker one now. You saw something coming out, right, from her abdomen? What do you think? She defecated, or has anyone seen that behavior before? No? Okay. So that's not defecation. So basically, if you want to learn more about it, I don't have time to go over that, but just Google for honeybee queen spraying behavior. Like you heard spraying in cats, right? They have territories and all like animals do. So this is something very unique in honeybee queens. They do this spraying behavior. So that chemical is very attractive. The thing that she was spraying, technically that the purpose of that spray was to hit the other queen. But this was a lab bench and she couldn't focus very well. So that chemical that is coming out of that compound is very attractive to worker bees. That's the hypothesis. So if the queen, the one that is a little dominant, if she sprays on the other struggling queen, then the workers will come and ball the queen, the one that is struggling, and they will suffocate and kill. So they will aid the other queen die. So that's again a very evolutionary process that people have hypothesized. There's no solid evidence for that, but that's hypothesis. But people have, at least there is one paper. If you read, I think David Tarpey, actually he did his PhD from UC Davis, and now he's a professor at. He's one of my colleagues in North Carolina State University. So he has done some work. Maybe he was in California when he did this work. I don't remember now. So he has one paper. If you just Google with David Tarpey, Tarpy, spraying behavior in honeybees, you will find that article. Read it, and again, you can find. Email me again, and I can send you a PDF of that if you want to read. The very fascinating behavior. So, okay, we'll switch to nutrition, and just bear with me, maybe another 7 or 8 minutes, and we'll finish this. So we have all know, right? We are you are what you eat. With human nutrition, we know so much. It's the same thing with bees, right? For any organism, nutrition is the first line of defense. It helps with the immune system, detoxifying enzymes, so many things. So on the left, I am saying macronutrients, which is your carbohydrates, right? Your honey, which is a nectar source or a honey source, that's your carbohydrates. But protein is really important. And that's what the pollen is, right? They don't get protein from anywhere else. It has to be your pollen. And then we know a lot about macronutrients, right? You probably know you need this much honey to survive the winter in California, wherever you are. Uh, Whereas micronutrients are the ones where we only have barely scratched the surface. We don't know much about what vitamins our bees need, what minerals, and especially these lipids and sterols are the ones I'm going to talk a little bit more today. These lipids are really critical for insect development, not just honeybees, for many insects. And this is where we have a big gap in knowledge. So again, a recap here, amino acids, crude protein comes from your pollen. It's a primary source of protein. 10 to 40% is the range. So don't think all pollens are same. They may vary a lot between each pollen. So that's why it's a beautiful evolutionary process. If you sit next to your hive, especially you're a new beekeeper, watch those bees coming in. They'll come with 5, 6, 7 different colors depending on the time of the year, because they want to balance those nutrients that are absent in the other pollen, because not all pollens are equal. So that's the beauty of collecting multiple pollens and mix. Your bees are very good at detecting concentrations of nectar. If you put 10 different petri dishes in your kitchen and allow some bees to come in, or put in your porch and try this experiment, so you will see a lot of bees coming, and they will be randomly on all those 10 petri dishes with different concentration. But eventually you will see more bees on the highest concentration of sugar, because they have good receptors for detecting sugar concentration. Whereas pollen quality is hard for them unless they have prior experience with some pollen. They may have some bias based on the phenolics. But other than that, if there is, your bees have never seen some pollens before, and you know the concentrations because you tested them in the lab, and you put 10, 20, 30, 40, 50% concentrations of these pollens, they will be all over the place because they can't detect the concentration of protein. But they are very good at detecting the concentrations of nectar. That's the other reason why you see them collecting a lot of different pollens as well. And nutrition is really critical when they raise those winter bees, which we loosely call them winter bees, but they are, a scientific term is diutinous bees. That means long-lived bees. So the bees that are produced in late fall are the ones that survive the entire winter, right? They are there until the fall comes. If you live in a cooler climates, then they may survive even 5 months. So those bees are really critical, and that's when you have to be very careful when the bees are raising those winter bees. You don't want too many mites on them or too many nutritional problems. Otherwise, you'll have a compromised bee in terms of physiology because of mites and then also because of poor nutrition. So those bees will not survive. I get calls from Oregon beekeepers. They say, my bees were, these are very new beekeepers. They're still learning the game. So they say, in October I checked and the bees were really thriving. I had almost 18, 19 frames of bees. I thought they will winter well. And then by December, all the hive collapsed. And that's because the bees will look good to you, right? Just physically, they will look good, but they have been compromised with Varroa and nutrition. Maybe they were raised in that environment where both Varroa and nutrition was a problem. Those bees' immune system will be so poor that they will not survive more than 2 or 3 months. So they will all die in winter. So most of those losses happen because of Varroa and nutrition as well. So here you can see those, I know this is not a good example for that, but this is what happens. So you see this bee on the right, if you dissect and look in the abdomen, you see all this fat body here, all the white stuff. That's what your bee should be looking in winter, before winter in late fall, because bees are storing everything they eat because there is not much larva now in October or September, wherever you live. There is little less larva when compared to June or July. So bees are still eating protein, right? Pollen. So they're storing more in their bodies and not providing much to the larva because there is no need. So that's how they deposit all the fat bodies and then they get to survive the winter because of high vitellogenin, which is a glycolipoprotein and all that stuff too. So sterols are really important. As I said before, they are important for the cell membrane development, molting hormones. I don't know if you remember from your high school, if you're not a biology major. Uh, Molting is really critical, right? So, and Ian is laughing. He's a teacher, probably. He has explained all these things before. So molting is a process like you'll see in your reptiles when the snake sheds the skin, something similar, right? So they have to shed their cuticle as well when they are growing. So the molting hormones are really important in insects. Without that, they can't grow. A larva, if there is no molting hormones, it will stay in the same stage. 1 day old larva will just be 1 day old. It will not grow from there. So those molting hormones are really critical. And for those molting hormones, these are precursors, the sterols, the 24 methylene cholesterol that I've listed here. It's a really important cholesterol for honeybees. And I know we talk about human cholesterol, we think our cholesterol is bad, right? Sometimes, but you need cholesterol. That is really important. Too much cholesterol, probably we don't. That's why we use all this anti-cholesterol medication sometimes to reduce our cholesterol levels. But cholesterol has really important functions of cell membrane development. There is immune function as well. So there's so many things. So your honeybees have something called 24 methylene cholesterol, which is the predominant sterol, and you need that in big amounts, and that comes from pollen. So when you feed your protein supplement, it doesn't have, it has only crude protein, right? You don't have any of those. That all comes from pollen. This is the hormones that I was talking about, ecdysone and juvenile hormone. So there is a beautiful balance between these two. Juvenile means young, right? So when the juvenile hormone goes up, the larva stays in that same age. It will be 1 day old for a while. And when the juvenile hormone goes down, then ecdysone goes up. And then you switch to the second day. So that's how this switching of different stages happen. And then you become, from an egg, you become an adult eventually, right? That's how it happens. So I'll skip this. I don't know if you want to watch this. I'm sorry I said I will take your time, but am I still okay, Ian? Or I can stop whenever you guys think it's too late. Can I go 5 more minutes?
You're good. Yeah, go 5 more minutes. No problem.
Okay. Yeah, I think I'll not need more than 5. I'll be quick. So you see that bee was eating pollen. So she got all those pink dots, which are sterols. Again, they don't look pink, but just for the animation. So the larva gets the sterol and then it becomes a normal bee. But just example, I'm not saying protein, don't get me wrong. Protein supplements, you need to rely on them when you don't have much going on in terms of pollen collection. But you see, most of the pollens, you don't have any sterol. It's only crude protein they're getting. So when bees are just fed on protein for a while, they will not have the sterols. So you don't see that pink dots transferred to the larva and the larva doesn't get any sterol. So it just shrinks and dies inside those cells. And the bees will either cannibalize or remove it from the cell. So molting hormones are really important. The take home message here, molting hormones are really important for development of any insect, not just honeybees. And then you need pollen in case you have honeybees for getting that precursor that you need for molding hormones. It comes from that sterol, 24-methane. So again, I'll not bore you with all the study designs here, but we have published a lot of papers on this 24-methylene cholesterol because in studies in 80s and 90s, they did figure out that this is the important sterol. But after that, no one studied to look at what is the optimal concentration you need in a bee diet. So based on this study, again, this is available online, I think free. You can just Google with my last name, Sagili, and say 24-methylene cholesterol, or if you can't find, I can send you to read. So we figured out it's only 0.5% of the total dry weight of the diet you need. It's not huge concentrations. So again, all this work that we are doing with micronutrients is the goal is eventually to improve the quality of the protein supplements that are sold in the market so that they are, I don't think we will ever get close to a pollen diet. What pollen provides, you can't mimic it in a protein diet, but the goal is to get as close as possible by understanding the role of micronutrients and the concentration that you need. So we are testing a lot of protein diets in the lab as well. You see, these are 3 different diets here. This is one group, this is one group. So we know that beekeepers spend a lot of money. I know backyard beekeepers probably not so much, but commercial beekeepers rely on this protein diet a lot. Some people feed here in Oregon, I would say probably even 15 pounds of protein supplement in a year. And that's very expensive. So we want to really understand, are these protein diets really doing something for the bees, or are they just ingesting and defecating and nothing is happening? So we are doing studies in a very micro level where we are trying to understand the gain in body weight, looking at the survival, looking at the digestion, looking at the assimilation of these diets. So you see on the right side, these are bees in a cage where we give them water, sugar, and then there's a pollen diet which you can't see really. That's the one where it goes in, and then the bees feed on those different diets. Again, maybe next time I'll give you more information. I didn't put too much information here. We have another interesting project going on, looking at the pollen nutritional composition. Probably as beekeepers, you are curious, or someone asks you, give me 5 top plants or 10 plants that I can put in the backyard that are good for bees, right? It's easy. You can come up with some names for sure. I can give you a few names now as well. But have you ever thought how these numbers are coming from? Like, or the choices are coming from? These are based on attractiveness, right? So someone has done, okay, lavender, I see 100 bees in a minute. I have borage, I have 70 bees in a minute. I have something like Phacelia, I have 60 bees per minute. So I think people have looked at attractiveness for sure, but I don't think people have looked at the pollen composition of those pollens to say this is best for your bees because this has this much crude protein, this has this many lipids, this has amino acids, this has omega-3 fatty acids. There are so many critical micronutrients that your bees need. But I don't think that type of information is available. That's the whole goal, what we are doing here with this USDA funded project is looking, it's a painful one. We have to collect pollen from a lot of, so there are citizen scientists involved like you guys, if you're interested in some pollens in your area, which are very unique, you think you can send me a gram of pollen, which is a very painful exercise to collect 1 gram. It looks a very small amount, but try collecting from some species. It will take days for you, thousands of flowers. So, so this is a lame example I'm giving you here. So most of the plant species that people try to select, I'm not saying that's wrong. Maybe that's a good way to do now, but wouldn't it be great to select in a more scientific way based on the pollen composition of those plants? So that's what I say here at the bottom. Current basis for selection is attractiveness based on whether bees come. So again, this lame example of French fries and salad here, if you give me both together, I might choose to taste French fries first, right? Because it's attractive. That doesn't mean that's the best food source for me. So again, not a great example. I'm just giving a lame analogy to get your attention. But that's what it is. Attractiveness doesn't always translate into a quality of food, right? So that's what my or better nutrition. That's what my term was. So that's what we are doing the study. So there are so many plant species. If you go to this USDA NRCS website, beautiful list of plants. You go to Xerces Society, which is in Portland here. So our goal is to provide another column here where you can, several columns actually. So where as a beekeeper or a conservation group or whatever you are, you go and you want to plant something like, okay, I want something like Canada goldenrod. You go and look at all these things that are there, depending on what region you are and all that stuff. And then you go back here and say, oh, based on crude protein and lipids and other thing, it doesn't look that great. You go to another choice. So that's what we want people to use this information eventually when it's available, is make a more judicious or a more a choice based on more scientific information rather than just attractive. So these are some methods we have done. Yeah, it took us a while to really figure out. We designed even a vacuum where you can suck up the pollen from some species with different mesh sizes, because pollen comes in different sizes from anywhere between 20 microns to 200 microns. So again, I'll not tell you that story, but this is what we envision. We want to get all those data like this and then put that in the website where those things are. Just one more minute, I'm done. So take your take home message. If you have access to adequate pollen throughout the brood season, that's what you have to make sure. I know probably some of you have no problem. You see pollen coming even late in fall. But if you live in an area where there is a pollen dearth even for a month, then always think about using protein supplements. Otherwise, your colonies will decline in size. They will not have enough population to survive the winter. So always try to use pollen supplements, protein supplements that have at least 5 to 10% pollen. Then some companies mix pollen and sell you. I think those are better than, they are maybe expensive, but then I think that's what the sterols and other things I mentioned. You don't need tons of those, but still having some pollen mixed in protein is always helpful. And as I said before, nutrition is really critical when they are raising those winter bees. So make sure that they have adequate nutrition. And some people ask me how to trap pollen, so I put this slide as well. You can collect your own pollen in a time when bees are bringing in. Don't stress your bees by putting traps when they're themselves struggling to get pollen. So maybe if you have a strong hive and you think a lot of pollen, just you can sit next to the hive and see. If you're getting probably 50, 70 pollen foragers in a minute, maybe that's a great hive. Then you can put these front porch traps and then you can collect pollen and you can store it in your freezer. And we have shown that pollen is probably a little bit deteriorates in protein content, but they're pretty good for a year. If you store in a Ziploc, put it in the freezer, and that pollen you can feed them next year to your bees by crushing them and putting in the protein supplement. If you're making your own or buying a protein supplement that does not have pollen, then you can, and you don't need too much. You can just put 2 tablespoons, maybe max. That's what they need. They don't need too much of that. But then they will get their natural flow again, right? So that should take care of. But there are some tips. You have to seal all entrances. Bees are pretty good. They will sneak in from other entrance if you have these auger holes or something. So you seal all those, and then you have to train them to get as well. Just don't go randomly and put this in the middle of the day. Then the bees will be confused because they have to go through this mesh here. If you can see my cursor on the top. So this actually flips. You can fold it back and put a tape on it and then make that entrance open. So the bees will back and forth. And next day morning, once they're trained, before they wake up, you can go and activate the trap. So that means it comes back in position. So the bees will get through those holes and come out, and then they know the weird beekeeper has done something, and then they will get trained in going through those holes. And then otherwise they will get confused and they'll go to the neighboring hive. That happens all the time. If the hive is next door, you'll see a lot of drifting happening because they're confused about the entrance. So do all this meticulously and collect some pollen, and trapping no more than 2 days. Even if it's a strong hive, they will be impacted if you're continuously trapping pollen. Okay, there is a nutrition paper if you're interested. We wrote this for veterinarians because they're supposed to write prescriptions for antibiotics for bees now. And so this was a veterinary journal that was interested in some nutrition information, but it is good for beekeepers as well. Okay, and Ian mentioned that we have a master beekeeper program. If you live more closer to Oregon border, you can probably think about it. But otherwise, I think my counterpart there, Lina Nino, she has a great program in your state as well. Okay, thank you very much. And if you have questions, sorry I went over time again, but hope it wasn't too boring.
Thank you very much, Ramesh. Thank you very much.
I'll stop sharing and see if you guys have any questions. Okay, Jamie.
We know that bees need better nutrition in the fall to make winter bees so they survive. Is there any information that nature provides for them such that the fall flowers that we know are great, the Asteraceae family the goldenrod, the coyote brush, the California aster are in fact highly nutritious and better sources of nutrition for bees?
Yeah, no, great question. Actually, there is a project this was not funded. We wrote a proposal a few years ago. I know there is only a limited money, right? There are so many proposals they have to fund. So I understand, but sometimes this important don't get funded. So we wanted to see different times of the year. We want to trap pollen like continuously in a year to see if the quality changes by the time. But that's a great question you asked. Maybe there is a evolutionary process where the plants that are blooming, as you said, goldenrods and other coyote brush or something, maybe that's what, if you have some access, you can send those pollens. Again, I don't have a great answer for you, but that's an hypothesis which no one has tested. Maybe the plants that bloom late in fall, maybe they have a better lipid content because bees will need more lipids at that time. So we think maybe those plants that are blooming late probably have a better nutrition in terms of especially lipids, and the sterols are lipids that I talked about. So again, those things have not been really studied well. So that's the reason I can't really tell you whether, but yeah, I think someone will do in the next few years for sure. So then probably we'll have an answer. So, but that's an assumption. We think maybe bees don't really seek protein. I think only in bumblebees, I've seen some studies, they say that bumblebees have the ability to select high quality pollen, but honeybees, I have not seen a study that has shown. Like that's what I was telling you in the example, right? I was saying you that we are trying to, if you put 10 different concentrations of sugar, they are able to distinguish sugars, but protein quality, people have shown they are not able to distinguish between different proteins. So I think they are still randomly collecting what is there available during late fall. Hopefully that is in nature, that's the best part available at that time, and they're still benefiting from it. Other questions?
I think you mentioned that professional beekeepers are feeding 15 pounds per hive.
Yeah.
So
Isn't
basically
it
full-time feeding.
surprising, right? Yeah. Because again, you as a backyard beekeeper, you will not need, because like an average Oregon beekeeper does 5 different crops pollination, right? Some are really bad. Have you heard of hybrid carrot seed? So in Madras, there is, I don't know how much you guys travel to Oregon, but in Central Oregon, across the mountains, on the Cascades on the other side, there is 5,000 acres of hybrid carrot seed production. So they boast that 70% production, 70% of world hybrid carrot seed production is in Madras, one location in Oregon. And that is a hybrid care. Most of the hybrid crops are not good for bees because in that process of plant breeding, they have messed up with so many characteristics that are good for bees. So they don't produce enough nectar, they don't produce enough pollen. So when a beekeeper goes to Central Oregon for pollinating hybrid carrot seed, those bees really suffer. There is not much there. So the beekeeper provides probably 6-7 pounds just in that one crop. So it depends. That's why you're probably amazed that how can someone feed 15 pounds, because they do pollination, right? So some crops are good. Almond actually is a good source of pollen. You probably know that, right? They come with tons of pollen when they move back to Oregon. I see 7 or 8 solid frames. Almond doesn't produce much nectar, but it profusely produces pollen. It's a very good source of pollen. We have tested the concentration of protein. It's about 29% of crude protein in almond pollen. So it depends on the crop. So they don't feed much when they're in California, probably a little bit in the beginning, but then they start feeding when they go to blueberry pollination. So it all depends on which crop they are in. So yeah, on average, I have seen some Oregon beekeepers about 15 pounds. It's a major expense for feeding as well. Any other questions?
I have another question about sterols. I just read an article that a lab, the Kew Gardens, they use CRISPR technology and they put it into yeast and they made certain critical sterols for that bees use in their nutrition. And then they were able to show that obviously you had better results when the bees used those sterols. My question is that supplemental sterile feeding critical, or is just a little pollen in the pollen subs enough? Do we actually have to supplement those targeted sterols?
Yeah, no, great question. Looks like you are keeping up with the literature more than me. So I just read that paper 3 days ago. So yeah, it's from UK. I know the researcher. Her name is Geraldine Wright. Yeah, I have visited her lab in UK 2015. So she does a lot of nutrition research like we do here in Oregon. Yeah, so for those of you, what Jamie is saying is, so they have genetically engineered a yeast species to produce this 24-methylene cholesterol that I was talking about, that your bees need. So they are trying to produce with this yeast. And so eventually their goal is someone will make that yeast that is high in 24-methylene cholesterol, and you will feed them in your protein diet. So coming back to your question, Jamie, I think as I said, you need only a small concentration. I said 0.5% of the total diet they are eating, right? So hopefully it's not that much. So this is a new development. It's a Nature journal. I know it's a very prestigious journal they published. I think they published in that journal because of the technology. It's not about the cholesterol itself, but I think the way they engineered the yeast, I think, is the major development there. So yeah, I think it has value. I don't know when that will become commercialized and if they will ever get it to feed your bees in a protein supplement. But coming back to your question, yeah, if you can, as I said, you collect your own pollen, if it's a good source of sterol, like borage is a very good sterol source as well. So if you can mix some of that, I think you can still do the job. But if they are selling you very cheap that yeast, maybe, then yeah, you can try that and help your bees. But I don't think you need tons of that sterol. So, but it's a really new development, and yeah, I'm glad you're reading those papers. Other questions? You want to watch some more videos? Okay. No, I know it's late for you guys too. So yeah, thank you, Ian, and yeah,
Thank you very
feel free
much.
to email me. I can answer your questions. You know, you may have some thoughts that may come later, but yeah, anything with nutrition, Varroa, yeah, even in general with bees. If you have any questions, email me and I'll be able to answer.
Thank you very much. Thank you for joining us.
Yeah, sure. Have fun, and yeah, hope good luck with your bees and everything else that you do.
Thank you. Good evening, everyone.
Thank you.
Thanks.
Thanks.
You have a good evening.